Students, hello and very warm welcome. Today I will be talking about the genus Anthoceros. The name Anthoceros means flower horn and refers to the characteristic horn-shaped sporophytes that all hornworts produce. The genus Anthoceros was first established by Lee as early as in 1729 and later was adopted by Linnaeus in 1753. As far as the systematic position of this genus is concerned, it belongs to family Anthocerotaceae, order Anthocerotales, class Anthocerotopsida, which belongs to division Bryophyta. Anthoceros is global in its distribution, with about 250 species in temperate as well as tropical regions of the world. Over 25 species of this genus have been recorded from India. Among Indian species, Anthoceros crispulus, Anthoceros erectus, and Anthoceros bhardwajai are commonly found in all the major biogeographical regions of the country, which include Eastern Himalaya, Western Himalaya, and South India. Now let us discuss something about its thallus structure. The plant body is a small dorsiventral dark green prostrate thallus, which is variously lobed with a tendency towards dichotomous branching and forming orbicular or semi-orbicular rosette. The thallus is several layers thick but lacks a definite midrib. On the undersurface of thallus, cavities are formed due to breakdown of cells and their replacement by mucilage. With the maturity of thallus, the mucilage in these cavities dries out, resulting in air-filled chambers which are later occupied by the cyanobacterium nostoc. As far as the anatomy of the plant is concerned, each cell of a thallus consists of a single large chloroplast which encloses several pyrenoids. The nucleus lies opposite to the chloroplast, usually near the pyrenoid body. I think it is pertinent to mention that the presence of chloroplasts containing pyrenoids is characteristic feature of alga, but is unknown in Archegoniaceae, the only exception being Anthoceros, Isoetis, and Selaginella. On the ventral surface of the thallus, there are many rhizoids, which are unicellular and smooth-walled. Tuberculate rhizoids and ventral scales are totally absent. The growth of the thallus is due to single wedge-shaped apical cell or a marginal meristem, where each cell has 4 cutting faces. Now let us try to know something about its reproduction. Anthoceros reproduces by sexual and asexual means. Asexual reproduction usually takes place by tubers, which are able to withstand unfavorable conditions. Other means of asexual reproduction include fragmentation, gemma formation, persistent growing apices, and apospory. As far as the sexual reproduction is concerned, most of the species of Anthoceros are monoecious, which include Anthoceros fusiformis, Anthoceros crispulus, Anthoceros golani, Anthoceros longi, and Anthoceros punctatus, while some species are dioecious. For example, Anthoceros angustus, Anthoceros chambensis, Anthoceros pearsoni, Anthoceros holli, Anthoceros laevis, Anthoceros khandaliensis, and Anthoceros erectus. Now let us discuss the sex organs of this genus, which includes the male sex organ known as antheridium and the female sex organ called archegonium. The antheridia develop endogenously from a surface cell which has denser cytoplasm and a conspicuous nucleus. It divides periclinally into an inner segment and an outer segment. The former functions as an antheridial initial and develops into an antheridium or antheridial cluster. The outer segment is the roof initial, which forms the two-layered roof of the antheridial chamber. A mature antheridium is an orange colored club shaped structure with multicellular stalk and cylindrical body, which finally forms anthrozoids. The liberated anthrozoids is a tiny biflagellate structure. The two flagella are equal or almost of the same length as the body. They are inserted at the slightly broader anterior end of the slender body, which is slightly curved. Just behind the front end and beneath the two flagella is an elongated swelling, probably a blepharoplast. On the contrary, an archegonium arises from a dorsal segment of an apical cell in its close proximity and remains embedded in the thallus. New archegonia arise in acropetal succession. The archegonia develop from dorsal segment of the apical cell. The archegonial initial cell becomes evident due to its dense cytoplasmic contents as compared to the adjacent cells. The primary archegonial cell divides by 3 vertical walls which intersect each other, forming other 3 peripheral cells enclosing inner axial cell. The axial cell divides transversely into an outer and an inner cell. The outer cell further divides transversely, forming primary cover cell towards the outer side and primary neck canal cell towards the inner side. The primary cover cell divides once or twice, forming 2 to 4 cover cells, while the primary neck canal cell divides transversely to produce 4 to 6 neck canal cells. The inner cell is the primary ventral cell, which also divides transversely, forming ventral canal cell and egg. A mature archegonium has 2 to 4 cover cells, a jacket enclosing 4 to 6 neck canal cells, ventral canal cell and an egg. At the time of fertilization, the cover cells are thrown off, the ventral canal cell and the cells of the neck disintegrate, releasing a mucilaginous mass which becomes continuous with the mucilage mound on the thallus surface. The sperms caught in the mucilage reach the egg and fertilize it to form the zygote. The first division of the zygote is longitudinal. In this way, hornworts differ from rest of the bryophytes, where the first division is transverse. Final development of the embryo results in the formation of sporophyte. After syngamy, the fertilized egg increases in size, filling the entire archegonial venter. Usually it divides vertically, but in case of Anthoceros crispulus, the first division is transverse. The second division takes place at right angles to the first division, forming 4 to 6 celled embryo, in which the cells are arranged in 2 tiers. Then it divides vertically, forming 8 celled octant embryo. Sometimes one tier may divide transversely, forming 3 tiered and 12 celled embryo, as in Anthoceros angustus, where the lower tier forms foot and upper 2 tiers form capsule. In case of Anthoceros crispulus, both the tiers divide transversely, forming 4 tiered and 16 celled embryo, in which lower 2 tiers form the foot and the upper 2 tiers develop into the capsule. The 4 cells of the uppermost tier, which are destined to form the capsule, divide by periclinal walls. This separates a central group of the 4 cells from the surrounding peripheral ones. The former constitutes the endothecium and the later amphithecium. From the entire endothecium originates the sterile columella. The corresponding tissue in the liverworts forms the archisporium. The archisporium in Anthoceros is formed from the inner cells resulting from periclinal division of the amphithecium. The outer cells of the series function as jacket or wall initials. The jacket or wall initials undergo repeated divisions to form the capsule wall. The outermost layer of the capsule wall is epidermis. They, as a rule, have 2 large chloroplasts per cell, sometimes 1, rarely as many as 4. This photosynthetic tissue communicates with the exterior through the stomata. The single layered archisporium is parallel to the elongated axis of the capsule and is dome shaped. It overarches the rounded apex of the columella and extends nearly to its base. In some species, the archisporium remains single layered throughout, for example, in Anthoceros erectus, and functions as the primary sporogenous layer. In other species, for example, Anthoceros himalayensis, it becomes 2 layered above the base of the capsule. In still others, it becomes 2 to 4 cell layers, thick by periclinal divisions, and forms the sporogenous tissue. The sporogenous tissue differentiates into 2 kinds of sister cells, namely fertile cells and sterile cells. The former gives rise to spore mother cells and the later to pseudoelater mother cells. As far as the spore mother cells are concerned, they are large cells oval to spherical in form. Each cell contains granular, denser cytoplasm, a distinct nucleus, and a chloroplast. The young spore mother cell increases in size, separates from other cells, and becomes spherical in form. Finally, each of these undergo meiosis to form the haploid spores or meiospores. This process is called sporogenesis. In this process, prior to meiosis, the single chloroplast in each spore mother cell divides into 4 chloroplasts. They separate and move to the periphery of the spore mother cell and become spaced equidistantly from each other. The final position of 4 daughter chloroplasts in the spore mother cell indicates the location of the poles of the meiotic spindle in it. The diploid nucleus at this stage undergoes meiosis. The resultant 4 haploid nuclei move to points already occupied by the chloroplasts. At this stage, cell walls are laid simultaneously between the 4 haploid nuclei, dividing the spore mother cell into 4 cells arranged tetrahedrally. These are the young spores. As the spores in the young spore tetrad ripen, their walls thicken. The spore coat in the mature spores is irregularly thickened. It is yellow in color in some species and black with some small tubercles in others. As far as the pseudoelater mother cells are concerned, they are slender, sterile cells with smaller nuclei. These increase in length but do not divide. Finally, they become united to form a sort of a network. In their intricacies lie the spore mother cells. Towards maturity, the network breaks into small, simple or branched chains of 3 or 4 cells. These are the pseudoelaters. The later usually lack the spiral thickenings characteristic of elaters, but are hygroscopic in nature. The apical growth of the capsule ceases with the establishment of archisporium, columella, and the wall regions. Further growth is by the activity of the basal intercalary meristem in the median zone. It continually adds cells at the base of the capsule. They become progressively differentiated into the columella, the archisporium, and the capsule wall. Consequently, the growth and the dehiscence of capsule extends over long periods. The sporophyte of Anthoceros continues to live as long as the thallus lives. It is a feature in sharp contrast to the short-lived sporophytes of the liverworts. Early growth of the sporophyte is accompanied by an upward growth of the archegonium and adjoining gametophytic tissue. The surrounding sheath is sometimes called calyptra, but involucre is a more appropriate designation for it. The calyptra is carried as a small cap at the top of the sporogonium, and the involucre forms a tubular sheath at the base of the mature sporogonium. This sporophyte is an elongated structure with budging foot that penetrates the thallus and an intercalary zone with meristematic activity and a cylindrical, smooth, elongated and erect capsule. At maturity, the cells of the capsule differentiate to form spherical or oval spore mother cells and elliptical to rectangular elateral mother cells, which subsequently result in the formation of spore tetrads and vermiform elaters respectively. If we analyze the foot of sporophyte, it is deeply embedded in the thallus and derives nourishment as the cells are in close contact with thallus cells. The region of the contact between foot and thallus tissue is well marked in many species. It is called placenta. The cells of gametophytic side of the placenta are called transfer cells. And if we analyze the structure of capsule, capsule is elongated, cylindrical, smooth, slender and erect. It is nearly of uniform thickness throughout its length except towards the apex, where it slightly tapers. Usually it is 2 to 3 centimeter, or in some species up to 15 centimeter long. It is light green at first, but turns brown towards maturity. The sporogonium or capsule of Anthoceros is capable of unlimited growth in length because of the presence of meristematic zone at the junction of the foot and the capsule. The apex of capsule represents its oldest part. The maturation of spores in the capsule is from the apex downwards. The capsule usually dehisces by 2 longitudinal lines to release the haploid spores. The spore germinates and subsequently develops into a new gametophyte. Now, let us conclude the topic by discussing the overall life cycle of the genus. The life cycle of Anthoceros consists of 2 regularly alternating generations, which include the gametophyte generation and sporophyte generation, with meiosis and fertilization as its 2 critical components. Gametophyte generation or the haplophase starts with the formation of spores. The spores are differentiated from the spore mother cells. They germinate under suitable conditions, each forming a new gametophyte thallus. The gametophyte is green and independent throughout its life. The gametophyte at a certain stage bears the sex organs, which include antheridia produced in antheridial chambers and archegonia, with 4 to 6 neck canal cells and a ventral canal cell and obviously an egg. All these structures produced by and connected with the gametophyte thallus constitute the haplophase or gametophyte generation with 1 N chromosome number. As far as the sporophyte generation is concerned, the sporophyte generation starts with the fertilized egg or the zygote. It has a diploid number of chromosomes. By active segmentation, the zygote forms an embryo. The later, by differentiation of cells and continued growth, develop into the alternate vegetative individual called sporogonium or sporophyte. It produces meiospores and aids in their efficient dispersal. The pre-spores are scattered by the air currents, which then develop into new gametophytes. The 2 phases of the generation constituting the life cycle of Anthoceros regularly occur one after the another. The life cycle of this type, which is characterized by alternation of generation and sporogenic meiosis, is termed diplohaplontic. With this, I conclude. Thank you.